Good afternoon, everyone. My name is Bangar Vaibhav, and I am from Latur district of Maharashtra. I have completed my post-graduation in genetics and plant breeding from RAK College of Agriculture, Siror. Today, I am going to teach an interesting topic: sexual reproduction in flowering plants. So, in today's lecture, we are going to see the structure of the androecium. After that, we will see the structure of the anther, followed by microsporangium, and at last we will see the structure of the pollen grain. So this is the lesson plan for today's lecture. So we will start with the lecture. We see the flowers in our day-to-day life. The shape of the flower, the size of the flower, and even the color of the flowers vary from species to species. These flowers are of great importance in our day-to-day life. They have the aesthetic value. They are used in ornaments, ornamental purpose. They are used in various cultural programs. Even our feelings of our human beings, such as the feeling of love, is expressed via these flowers. So, if we talk from the biologist point of view, these flowers are the sexual reproduction organ of the plants. This flower consists of two essential whorls. These essential whorls are also called as reproductive whorls. The first is the androecium, which is the male reproductive organ, while the second is the androecium, which is the female reproductive whorl. This male androecium, this male reproductive organ, androecium, it is made up of the stamens. It is the collection or it is the whorl that is made up of the stamen. Now this stamen, it has two parts. First is the anther and second is the filament. This filament is a long slender-like structure, while the anther is the bilobe structure found. We will see this anther and this filament with the help of the diagram. So in this diagram, I have drawn the filament. This is the long filament, long slender structure that is called as the filament. While this upper portion, it is called as the anther. This filament, it is connected to thalamus or the petals of the flower with the help at the proximal end. At the proximal end. Now, if we look at this anther, we see here two lobes. This is the first lobe and this is the second lobe. So we can say that the anther here it is a bilobe, that is made up of two lobes. Now, if we see in each lobe there are two theca present. This is our first theca and this will be our second theca. Similarly, in the second lobe also there will be two theca, first theca and the second theca. So we can say that the anther is bilobe as well as it is dithecous. So the anther found in the plants of the angiosperms, it is bilobed and dithecous. Now, if you want to study the further structure of this anther, we have to take this transverse section. Now, I will draw the transverse section of this anther so that we can study the other parts of this anther. So, transverse section. This is the transverse section of the anther. Now we see these the four corners are present. These are in the shape of a tetragon. So at each corner there is a presence of microsporangium. At each corner a microsporangium is present. And this microsporangium they further develop to form the pollen sac. Now we have seen this structure of what is present in the transverse section. Now we will see what are the different types of layer that are present in the transverse section of the anther. So the outermost layer that is present is the epidermis. This is the outermost layer. The second layer inner to this epidermis, which I am drawing with the help of this red marker, is the endothecium. This is the second layer. Now the third layer, which again I am drawing with the help of black marker, it is called as the middle layer. This is the third layer. It is the middle layer. These three layers, epidermis, endothecium and middle layer, they provide the protection to the microsporangium, and they also help in the dehiscence of the anther. Now there is a fourth layer which is called as the tapetum. This layer, this is the fourth layer that is called as the tapetum. This tapetum layer, it provides the nourishment to our pollen grain. Tapetum, this is the fourth layer. Now if we look at the center of this microsporangium, we will see a group of tissues which are compactly arrayed, and these tissues are homogeneous in nature, and these tissues are called as the sporogenous tissue. Sporogenous tissue. This sporogenous tissue has ability to divide itself. So a type of division that is going to take place in this sporogenous tissue is the meiosis. Now if these cells of this sporogenous tissue are going to perform the meiosis, they will further convert into microspore tetrad. Now each cell of this sporogenous tissue is capable of becoming a pollen grain or the pollen mother cell. Now the process of formation of the microspore from the pollen mother cell is called as the microsporogenesis. I will repeat it again. The process of formation of the microspore from the pollen mother cell is called as the microsporogenesis. Now, when this anther matures and there is dehydration in this anther, the cells of the sporogenous tissue get separated and the pollen grain is formed. And with the help of after the dehiscence of these three layers, the pollen grain is released into the air for pollination purpose. So we have seen the transverse section of the microsporangium. After this, we will see the pollen grain, the structure of the pollen grain. Pollen grain. This pollen grain, it represents the male gametophyte. It is the male gametophyte. It is male gametophyte. It can be defined as it is the haploid cells that represents it is the haploid states that occur during the life cycle of the plant. So the pollen grain represents the male gametophyte. Now if you look at the structure of this pollen grain, it is a spherical in shape, and the size of this pollen grain, it is between 25 to 50 micrometer. The cells of the pollen grains are covered with two types of layer. The outer layer is exine, while the inner layer is called as the intine. I will draw the structure of these two layers for more clarification. This is the outermost layer, which is called as the exine. Now this exine, it is the most resistant organic matter that is found on the planet Earth. This exine, it is the most resistant organic matter that is found on the planet Earth, and it is made up of the sporopollenin. This sporopollenin can withstand the high acidic nature, and it can also withstand the high temperatures, due to which these pollen grains can be preserved as the fossils for much longer time. Now this exine has a small pore which is called as the germ pore. This layer of exine, it is not continuous. At one point there is a small gap, which there is a small pore which is called as a germ pore, and this germ pore will help for the further development of the pollen tube and the other processes that are necessary for the release of this pollen grain. So this was our first outer layer. Now the second layer that is present in this pollen grain is the intine layer. This intine layer is continuous, meaning that it do not have any type of pore, and this intine is composed of the cellulose and pectin. So we have seen the two types of covering that are present on the pollen grain. A mature pollen grain consists of two cells. First is the vegetative cell, and second is the generative cell. The structure of these cells also we will learn this with the help of the diagram. As I said, a mature pollen grain has two types of cells. First is the vegetative cell, and second is the generative cell. This will be our vegetative cell. This will be our generative cell. Now, this vegetative cell, it is composed of a dense cytoplasm, and it will provide nutrition to this our generative cell. While this generative cell, the nucleus of the generative cell, it is of irregular type and of irregular type. Now, when the development in majority of the angiosperm, that is 60% of the angiosperm, the pollen grain is released at the two-cell stage. At the two-cell stage, two cells that will be present are the vegetative cell and the generative cell. But in remaining 40% of the angiosperm, there will be the mitosis. Mitosis will occur in generative cell. Mitosis in the generative cell. In 60% of angiosperm, the pollen grain will be released at a two-cell stage, which consists of vegetative cell and the generative cells. But in remaining 40% of angiosperm, the mitosis will occur in the generative cell, which will give rise to the two male gamete. And the type of division will be mitosis. So, this is the structure of the pollen grain. I hope all of you understood today's lecture. So the remaining parts of this topic will be covered in our next lecture. Thank you.